report part A. The external examiners are about 2/3 of the way through. Hopefully they'll be finished soon. I might have to give them some help as well. Thursday at 1 o'clock, you'll see the next list of topics for report part B. You'll have a chance to look at those, peruse them, and you'll select them in the workshop labs next week in those sessions. I've seen a draft exam session, which I don't think you guys have seen yet. Yeah, what I've seen so far, which correct me if I am not correct, because it may change, but because this is a fairly large unit, nearly 400 students, usually it comes early, and what I've seen is Tuesday, the first Tuesday of the exam period. So you might want to start planning your attack of how many days you have left before the metabolism exam. Later on, we've got this, which is the last of what I'd call the grunt biochemistry, looking at pathways, and some things we need to know, some things we need to know the overview. The next 3 lectures will be integration. It's about how you control your brain with your stomach and your fat cells and vice versa. The final 3 lectures, so not next week, the week after that, they're all face-to-face. The last lecture, the review one, how does that go? Blackboard Collaborate, so we'll be on Blackboard Collaborate, and because that exam is in the first Tuesday of the exam period, hopefully it stays there, at least for me, it gives me more time to mark it. It gives you time to go into this session, so does your colleagues at Parramatta and Hawkesbury as well. Remember, in that week 14, there'll be another catch-up quiz. Okay? Remember, you have 12 quizzes, the 10 best ones count. And remember, the first quiz was a practice. I didn't have any marks on that because some people aren't here doing other things and we're just getting organised and all those kind of usual student issues. Okay, we've got a green light. Indeed, we do. Are we ready to get rolling? Is that a yes? Are we ready to roll? Yeah, you. Good. Okay, we're dealing with the citric acid cycle basically today and the electron transport chain. Now, remember, I know we get collective amnesia when we're students, going from one unit to another and having a break somewhere. Do you recall the last week before the break, we looked at a big enzyme and its acronym was? PDH. Remember that was in the cytosol of that cell? What did you make with that? It's in the mitochondria as well. You're making pyruvate, there it is, PDH. Pyruvate's got to move into the mitochondria. Now, I'm going to ask you to draw kind of a line around here through pyruvate, back down around, and everything underneath that line is in the mitochondria. What you've generated with PDH is acetyl-CoA. You have to get pyruvate into the mitochondria to do that. How do you do that? Well, we saw amino acids being cleaved. We saw fats being cleaved. Right down there, beta oxidation. Every time you cleave a carbon bond, you generate electrons and protons. Most of that's in the cytosol. Well, here you are cleaving the last one of glucose. You generate some electrons in the cytosol, but then you've got to take pyruvate into the mitochondria, right next to it. Pyruvate's got to get into the mitochondria. You'll see a transport protein taking it in later on today. Once it's in the mitochondria, our old mate PDH will convert that to acetyl-CoA. Remember, the CoA is just the carrier. That acetyl-CoA can now go into our citric acid cycle. Well, our citric acid cycle zooms around. There's our old mate OAA, right down the acronym. It's the same OAA we saw before. That OA can come back to make other things, as we've seen before. As you run around the citric acid cycle, you break carbon bonds, so what do you generate? CO2. Circle the 2 CO2s. How many carbons going in? Well, acetyl is 2 carbons, isn't it? You've hooked it up with citrate, so you've made a 2-carbon extra molecule. You zoom around the citric acid cycle, and what you do is cleave those carbons back off again, which makes no sense. That's what you're seeing over here to the right. What I want you to circle, that C2 is acetyl-CoA. C2 is acetyl-CoA. You've just made this C6 carbon compound. If you break a carbon bond, what do you generate? Circle that and write electrons and protons. NADH, your electron-proton carrier. If I cleave another carbon bond, I'm now going to go down to a carbon 4. I generate another electron and proton carrier and some more CO2. You're exhaling CO2. It comes from your tissue, but it's changed into bicarb before it gets to your lungs. Note, put a line through the middle of that cycle. That left-hand panel is where you break carbon bonds. You generate electrons and protons. What you do around here is generate this character, GTP. You're going to write down underneath that ATP equivalent because the phosphate's going to be donated. You generate some more electrons and protons to generate C4. What are you going to write next to C4? What acronym? OAA. That is the citric acid cycle. It's pretty straightforward. We're going to see some detail and some key carriers, and you don't have to memorise everything, but understand the principles. Half of it is getting our pyruvate in, the other half is cutting the carbon bond, and the last bit is regenerating OAA. So ask yourself, if OAA is used to do something else, what happens to your citric acid cycle? It slows down. What are you trying to do with this cycle? Generate ATP energy. So what do you do with all these electrons? You see them here, but right next to the electrons, protons as well. The electrons are going to move through this complex here. This complex, and you see those little closed pegs there, I want you to write inner mitochondrial membrane. That inner mitochondrial membrane has this pink thing, this yellow thing. It's got Q. We'll see who Q is later on. Another blue thing. There's cytochrome C. Haven't we seen him before? We see him a lot in advanced cell biology. It's bouncing around in the intermembrane space. I'll show you an animation of that. And another protein, complex 4. See that blue arrow zooming through there? I want you to write, that's the path of electrons. You are controlling those in your ETC. As the electrons move through and charge up those proteins, they pump out protons. So let's look over here, complex 1. As an electron zooms through there, gets transferred to Q, it pumps 4 protons out. As an electron comes into this system, I'll talk about complex 2 shortly, transfers its electrons to Q, it will help move more electrons through this system here, complex 3, pumps protons out. Those electrons you've zoomed through and you've kept them in the membrane as Q, get transferred to complex 3, and complex 3 transfers those electrons to cytochrome C. Remember the animation, we saw this little thing light up and it bounced to another protein. I'll show you later in the semester on review. Zooms across the intermembrane space and gives its electrons to complex 4. Electrons zoom through there, there's the blue arrow again. As it does, it generates water. Because of that event, it pumps more protons out. What you're doing here on this outer membrane of the intermembrane space is generating lots of positive charges. What you do inside the mitochondria is generate lots of negative charge. What have you got? A charge difference. What do the protons want to do? Come back in. That's why this membrane is rather unique. It stops protons coming in. The only way they come in is through ATP synthase and another protein we'll call UCP1. That complex system tells you one thing. If I did a gene deletion and remove complex 4, would you need to breathe oxygen? If I took your mitochondria, did a gene deletion of the complex 4 gene, what would happen to you? Sorry? You wouldn't be able to make water? Are you still pumping protons out? Look downstream, could you make ATP? Yes, you could. You breathing oxygen only generates 2 more protons on that space. But if we turn the oxygen off in this room right now, you won't last too long, will you? It tells you how demanding your cells are for those protons just simply to make ATP. After the break, we're going to see some molecules alter these pathways. Things like cyanide and arsenic screw up these pathways particularly well, make you dead very quickly. But those blue arrows are electrons, charging up the system, but also that's potentially dangerous for mitochondria. What I'd like you to note, even though it's not for this unit, cytochrome C is in the intermembrane space. If that molecule is loose in the cytoplasm because the membrane's been disrupted in the mitochondria, the cell's going to die in an orderly fashion. That's a way to tell how old the cell is, what's happened to it. But sometimes that disruption can be mutations because of these reactive oxygen species we're talking about. This pathway generates a proton motive force. Write it down now. Protons want to come back into the matrix. They can only get in through ATP synthase, but they can also come through and generate heat. And that heat generation is in brown fat. Is everyone kind of clear on that? Brown fat, not white fat. White fat's for storage. Why would brown fat be brown, do you think? Any ideas? Look at cytochrome C. What's its name? Cytochrome. It's a coloured molecule. Mitochondria look dark down the microscope. So if your brown fat was kind of dark, what do you think might be lots of in that brown fat cell? Mitochondria. They're not making so much ATP, they're making heat energy, which is what you've been coming through all winter. Today it's a bit warmer, isn't it? Feel a bit warm, start peeling off, but it's not really hot yet, is it? Not 37, 40 degrees. While you're in a cool phase, climate-wise, you've got more protein, we're going to call UCP1, to generate heat to keep you warm. As you transition to summer, you reduce that amount of protein, that UCP1, so you don't need so much heat generation. Is everyone kind of comfortable with this? So yes? Yes? Okay, I had a special request. Anyone know what parturition is? Oh, come on. Birthday. Write it down. Parturition is your birthday. It will be. Okay, let's get back to the mitochondria. It's actually useful to think about your birthday because when you're born, totally. I won't talk about it to stoke you or anything else, but when you're first born, you come in with all your clothes on, with a nappy on? No. What you have is a lot of brown fat, very active, down the back of your spine, down around your kidneys, to keep you warm for a start. That's very active in those first few hours of your parturition. So there was probably a lot of brown fat, hopefully not as much now. Let's get to the mitochondria. Let's get to the mitochondria. I know too much frivolity. Let's get back to work. And cakes for everybody, by the way.
Thank you.
You're welcome. Mitochondria. I want you to look at those first two headings. Your liver has about 800 to 2,000 in each cell. They are generally round. They can change shape, sometimes oval shape. They move around the cell. They're not stationary. Motor proteins move them around. They have 10,000 sets of ETC. What's that mean? Not etc. Just stop writing for a moment. 10,000 sets. They have 10,000 of these. That's a set. A set of the complex 1, 2, Q3, cytochrome and complex 4. 10,000 of them in your muscle cells to generate the ATP just to do the work. Now look at what's happening in your heart. Your heart has over 3,000 elongated mitochondria in each heart cell. It's still a myocyte, but that's constantly beating, isn't it? How many ET sets does it have? Over 30,000 to keep you alive, to keep pumping blood. The membrane composition, that's for some other units, particularly advanced cell and others, is important because the outer membrane has got around 30-40% lipid. The inner membrane changes significantly to keep the protons out. That's the schematic down here. The yellow convoluted thing is the inner membrane. That's the same thing you see in this cut-out version over to the side. That net charge there, I want you to write next to the negative, is negative proteins, negative molecules of all sorts. Not just chloride ions, amino acids, all negative charge. On the outside, you see that plus, that's the protons. Because of the charge differential, you're going to have those positives trying to get back in. The inner membrane, lots of protein there as well, but it has lots of cardiolipin. It does not allow protons through. Very difficult to manage, so we're trying to stain those cells. We can see all this convolution. What does convolution mean to you when you see somebody convoluting something? Increasing surface area, making it more available so we can move stuff around. The same thing when you have more elongate mitochondria. You've got more convolution, so you've got more ETC sets as you run through. Our outer membrane, we've got to get stuff through. Our inner membrane, we have the ETC sets, and I want you to circle ATP synthase. That's an integral membrane protein of the inner membrane. Now, some of these enzymes, like we've seen before, are actually embedded in the membrane as well. We're going to see those. What also happens in here? Beta oxidation. Didn't we take fats in with our carnitine, cleave them, and every time we cleave the bond, out comes 2 carbons, there goes some electrons. What do I do with those 2 carbons? I've got acetyl-CoA. What can they do? Go into the citric acid cycle. So we generate more energy. Can I move to the next one? Is everybody comfortable with this one? As you're moving on and your hands waving across, turning the page or typing the key, think about an athlete. What does an athlete do by training to the level of mitochondria? How many ETC sets? What are they doing, do you think? Increasing, indeed. If you stop active work, physical activity, what do you think you're telling your ETC sets and mitochondria? Stop needing them. You reduce the amount of energy. Is it immediate? No, it takes time. It's gene expression. It's hours and days. Think about the time frame to do that. It takes hours and days to build up muscle mass. Doesn't take too long to lose it, though, does it? Let's have a look at the citric acid cycle. I'll show you the little animation first, and then work back through the key steps. Okay, the citric acid cycle, you've drawn a little circle halfway through it, haven't you? Drawn a straight line through that circle. There you've got this left-hand side, you are cutting carbons. Now you see some delta Gs. I want you to ignore them for the time being. I'm not going to assess you on delta Gs. Here's our little animation, very few in this unit, unfortunately. We're going to run through this cycle and see what happens. We're going to generate electrons and protons. We're going to cleave carbon bonds off of our acetyl. Does that acetyl come from glucose, fat, or both? Which one? A, B, or C? C, write it down. Can come from any of those 2. Now that'll depend on the cell type, won't it? Would you expect liver cells to be taking fat into the citric acid cycle? Generally no. They'll be making fat if you've got excess carbon. Let's run around the citrate. What have you done? You've generated that compound. You've now cleaved a bond. That bond generates CO2. You've generated electrons and protons. There it goes in the bucket. That's your bucket list. Now you've got this thing, alpha-ketoglutarate. Haven't we seen that before? Very important molecule, isn't it? Part of the citric acid cycle. If you suck that out of the cycle, you slow it down. I cleave one more carbon bond. What have I got? Succinyl-CoA. Haven't we seen that before? There goes my CO2. In comes my electrons, protons into my bucket list. I've generated a little bit of ATP. I want you to write, oops, stop there, go back a tad for old people on technology. There, I want you to just note that ATP is a GTP product The GTP donates the phosphate to make ATP or ADP. So it's a donor. That's why it's called a GTP equivalent. What are we going to do now? Move this stuff around and generate more electrons and protons. Remember, those electrons and protons are in the matrix of the mitochondria. You're going to move those electrons, as we saw through the ETC, and then pump protons out. For a glucose molecule, 1 6-carbon glucose, we go around this cycle how many times? Twice, because it's a pyruvate coming in. What do you generate from that? A heck of a lot of electrons and protons, but not as much as you get from fat. What's important about this pathway? Well, I've circled in red the key characters. What does a red cross mean now to you? That's an inhibition, isn't it? What does that green triangle says? It means go forward, just like a green light. So who's saying do what? Well, pyruvate, we've just got into our mitochondria. What are you going to write next to that thing? Pyruvate dehydrogenase complex. PDH, write it down now. You can do that in an exam, which I wrote over semester break for you. This character here. Coming in, who slows it down? Too much ATP. Why are you going this? Why are you making this stuff? Don't. Slow down. Acetyl-CoA, slow down. A lot of AMP, as some of you discovered in the mid-semester, means your cell's got to be busy making more currency. CoA, NAD+, remember our friend calcium, circle it. It says we're busy doing something. It's released in the cell. What have I generated? Acetyl-CoA from either fat or pyruvate. What have I got here? Inhibition by NADH, CoA, citrate, ATP, slow down. Speed it up with ADP. Who does this work? Citrate synthase. Why have I got delta Gs? You covered delta Gs and FP and G a bit, didn't you? Didn't you? I was just talking to the unit coordinator this morning. Should have asked that question, shouldn't I? Delta Gs give us an idea of energy change. I don't like them in this unit because the cell doesn't worry about what goes on in a tube. The cell is too dynamic, changes too quickly. We're going to generate citrate. The next key step here is here with this dehydrogenase. This isocitrate now running to alpha-ketoglutarate. What slows it down? Too much ATP. What speeds it up? Calcium and ADP. Circle the ones that speed up the pathway. Put a square bracket or square box around the ones that slow it down to make sure you remember, because you're going to have to explain that in the exam. I run down to this guy, this thing, ketoglutarate dehydrogenase. I want you to write next to that complex. It's similar to PDH. Not exactly the same, it works similarly to PDH. This thing, alpha-ketoglutarate dehydrogenase complex. It's like PDH. Well, why is it like PDH? Well, it's taking alpha-ketoglutarate in this process to make succinyl-CoA. What have you put on that molecule? Ketoglutarate, circle the CoA. Didn't you do that up here? Put a CoA onto our 2-carbon in the PDH complex. I grabbed it, transferred it around, swung it around, and I stuck it on the CoA. You do it again later in this part of the pathway. There comes now our GTP. I asked you to write down the equivalent, didn't I? That's why you see the bracket for ATP. The phosphate from this can go to other places, including ADP, in fact. That succinate dehydrogenase, I want you to put a big ring around it. That's a membrane-bound enzyme. That's actually complex 2 of the ETC set. This character is complex 2 in the ETC set. We'll see in a moment. That's going to help transfer electrons and protons. It generates FADH. The intermediate step is malate, ultimately OAA. To complete the cycle, now people argue about this, to complete the cycle I need an OAA molecule. If I suck one out, I can't complete. It means I slow the pathway down. So I need to keep that process. Now it's often called regeneration, and there's some anapleretic reactions we'll talk about later on. Those red boxes are the key steps in citric acid cycle. I asked you to circle this one because that's a key protein, complex 2 of the ETC set. Can I move to the next one? Okay. And now we get to reality. This is when you go, something like that, yeah. Why am I showing you this? Well, because, I'm trying to simplify it here. I need to show you a bit of reality occasionally, otherwise you think it's too easy. And to make you feel sorry for me, because we had to remember delta G's, cofactors, all that. But what's kind of critical here is these double-ended arrows. What's happening over here? Well, if you look at the right-hand side and you draw a line down the middle, there's our succinyl-CoA. What happens over here? I've circled in here the dash line, and down here I want you to put one as well. There comes your CO2. Up until the point that you cut, it can go backwards and forwards. Once you cut off that CO2, you're committed to this thing, the ketoglutarate. You need that ketoglutarate complex to bring in our CoA. There it is. What have you generated? Succinyl-CoA. Once you've got that, you can go backwards and forwards a little bit. Same here for our fumarate. Same here for our process to malate. Same here for OAA. So these steps, and I again want you to circle this one. That's a one-way step. Down here, it's a one-way step. Another one-way step where you cleave the carbon bond. That's all I'm after with this slide for you. What I want you to note for the rest of it, it's reversible. But once we clip a carbon, once we generate, in this case, our OAA, we can either leave the cycle or we go back into the cycle again. That's the critical step for this particular slide. What's useful to think about is some of these cofactors we've seen before. Here's one, manganese, an active site stabiliser for decarboxylation. Now, I mentioned before, this country, as in the countryside, the soil is deficient in cobalt, magnesium and manganese. We often have to supplement those to livestock because they don't grow as well. They get ill-thrift. They don't generate much protein mass. You can feed them all you like, but no manganese, they don't generate the energy, they don't grow. That's a problem. Shall we move to the next one before we get more nervous? Okay. Are we right with this one? Not yet? The longer we're on this one, the more torturous it is, isn't it? Last day of grunt biochemistry. We right now? Is that a yes? Everybody else right? Okay. And it's funny we bring this up now because I just finished writing your exam and the last multiple choice question, number 60. Tell me which chemical structure this is. A, B, C, D, or I think it's only D I've given you. Who's this character? Base, sugar. Indeed, I was arguing with a chemist about this the other day. A couple of phosphate linkers, another sugar. Now I've got a ring structure here. This is my favourite energy carrier. It should be yours because it fluoresces. Put the right wavelength on, this thing fluoresces all over you, which is really cool to find out how it affects your gene expression in your nucleus because there's no mitochondria in your nucleus, particularly in your stem cells and your tumour cells. What does this thing do? This is NADH, and it might have a phosphate on, so it's NADPH. It's an energy carrier derived from a vitamin, like we've seen before. And again, you don't have to draw the structure. I've given you one. You tell me which one it is. What does this character do? It generates a couple of electrons, carries them as protons as well. Every time you have electron, negative charge, what do you need to do? Balance it with a positive charge. 2 electrons, 2 protons. But this character is going to give it up. It's going to give it up to the membrane and the ETC set. It can carry it around in the cell. It can take it to the nucleus and help with work there. So it's a way to manage high energy activity in a stable complex. That's what these energy carriers are. That's what I have down here, to stabilise it so that you can use it elsewhere. Now we can tell the difference when this molecule hooks up with somebody else. Yes, mate? It's funny you say that. I was just about to talk about that. NADPH and NADH have different fluorescence emissions, so we can actually tell who's who. and who's doing what. Now that changes when they're hooked up to different characters. So they don't just hook up to complex 1, 2, 3 or 4, they hook up to many proteins doing many things. And we can tell the difference now, some of my students can, about who they're hooking up to. The phosphate might mean that they're more involved in phosphate derivation in terms of what a protein does, which is what we're trying to work out. That's a very good question, and one we're still working on. Can I move to the next one? Just because it's his birthday, he thinks he has leave. If it was easy, everybody would be doing it, wouldn't it? As I said before, if I don't challenge you, who will? And if I don't challenge you, how can you be as good as you possibly can be? That's one way to think about it. Are we set to move on now? And as one of my postgrads love to say, no pressure, no diamonds.
So does this mean that everybody got one mark on the exam?
I will comment on the exam, but I'll save that till next week. Okay? I'm not going to give anything away just yet, sorry. And I don't play poker. Okay, can I move on now? Okay. What's this telling us? Don't write anything for a start, just look at it. In the middle, we have one of those roundabout things, you know, going round and round, citric acid cycle. What have you got? The key players. Incoming acetyl-CoA. There's citrate, there's ketoglutarate, there's CoA, there's malate, there's OAA. What have you coming out of this? The beginning of the semester where we started. We talked about other pathways that can link in and out of this core one. Who have we got here? Citrate. I write in there citrate shuttle, don't I? I would if I was you. Citrate shuttle, what do? I send carbons out, and what do I start making? Fat, through fatty acid synthase. I run around to ketoglutarate, what can I make from that? Well, we saw that so many times at the beginning of the semester. If it comes out of the mitochondria, I can make glutamate, amino acid. From that, I could make purines if I was desperate. I could make other amino acids like arginine, proline, and glutamine. Wasn't arginine important at the beginning of the semester? Who for? You, me, or your children? I run around to this character, succinyl-CoA. What do I make? Porphyrins and heme. I made a point of showing you how you make a heme function. And if you make it with so many enzymes, and one of them has a defect, you have a porphyria, don't you? That might be in report part B as a topic. I run around a few more steps. I get to malate. Can I get there from pyruvate? Yes, you can. He doesn't do much else unless I use the malic enzyme. I get to OAA, write down OAA. You can see how busy that character is. I can go into what? Citric acid cycle. If I come out through PEP carboxykinase, what have I made? PEP. Where am I? At the bottom of glycolysis. If I go up, put that arrow there, right next to it, gluconeogenesis, I can make glucose. Who does that? Which cell types? Cell type. Livers, hepatocyte, write it down. If I run this way, with our OAA, can I make some amino acids? Yes, I can, aspartate, asparagine, and I can make some pyrimidine bases if I'm really desperate. From my PEP, can I make other amino acids? Again, that's the beginning of the semester. That's why I started off in the periphery, danced around that, the pole. Think of that, pole dancing, me? That would be a nice thought, wouldn't it? Danced around that. What's the pole glycolysis? The bottom is citric acid cycle. Hopefully you remember that. So this cycle is important, but if you suck anything out of it, what do? You slow it down. It doesn't make ATP. So if you're deficient, and this is important for you in your diet, if you're deficient of any of these things, or if you need a lot of it for repair, specific amino acids deficient, what do? You slow down ATP. What does that mean? You're less energy productive. You can't do as much work. That's what it means to your metabolism. That's important for the next 3 weeks because this is inside the cell still. Now you're going to have hormones to tell you what to do, how much to eat, when to do it. Stress is going to tell you to be more active or less active. They're going to impact what this cell does. Here's the overall pathway. Now you notice this thing called anapleuritic reactions. What does that mean? You're trying to supplement what you suck out to try and keep those other pathways working. pyruvate with some bicarb and ATP through this carboxykinase, you've got OAA. Who makes that? Liver, and circle kidney when it's desperate. Remember, the kidney only starts doing these jobs when things get desperate. We'll see that in the next few weeks. PEP, CO2, GDP, so not all your CO2 is exhaled. With this carboxykinase, you can make OA. And notice they're double-ended arrows, depending on supply and demand. OAA, in this case, GTP. Who makes that? Heart and skeletal muscle. When would that occur, do you think? When the heart's really pumping, or when you're really working hard? And when you exercise, I have to tell this to people all the time, when you exercise, you should have a recovery phase. Allow the cells to generate the genes to make more protein accretion if you want to build protein mass, or try and change your gene expression to deal with that demand. But if you keep on exercising without a recovery, then you keep stressing those cells metabolically. Does that make sense? A recovery is important. PP with some bicarb to OAA. Who does that? Well, plants, yeast, bugs, who cares? I have to deal with those with a little project I'm running at the moment. What about this pyruvate, bicarb, this malic enzyme? Prokaryotes and eukaryotes deal with that when they're really important, particularly yeast, again. This pyruvate carboxylase is inactive without high acetyl-CoA. What does that mean? What does acetyl-CoA in a cell mean? Haven't you just cleaved it through pyruvate, through PDH to make acetyl-CoA? Can you go to the citric acid cycle? Yes, you could. It depends what your demand is. So you can actually speed the cycle up if you need to. When does that occur? When would you speed up the cycle? Are you speeding up the cycle now? A little bit, yeah, comes into play. Stress, one of them. You're all sitting here looking at me going to sleep, aren't you? Oh, it's been a week off, nice and warm. Do I have to listen to this stuff? Yes, you do. It's in the exam. But if I get you to run out because there's been some radiation leak somewhere from some silly character's lab, you'd race off like crazy, wouldn't you? What do you need? A lot of energy very quickly. Keep that in mind as we go through the next few weeks. Let's try and summarise that, shall we? The citric acid cycle. I've given you the Cook's tour so we don't have to go through every nitty-gritty detail. Just write down again, no delta Gs. There's none in the exam, none of the details. It's the overall concepts, the key players. 2 carbons enter the cycle as what? Acetyl-CoA, write it down. The acetyl function. They're not the ones removed during that cycle because it's like the citrate's the carrier but then cleaves it off. Our OAA molecules are not generated per se. You don't make them. You basically regenerate them because you started with OAA. So I know it sounds a bit odd. And you try and keep them roughly in cells of 1 micromolar. You notice the micro and not the U. The cycle is used to transfer electrons where to? The ETC, which we're about to get to, the details of that. This thing is essentially amphibolic. What did I give you at the beginning of the semester? The first slide, an overview. Catabolism, where you break things up, break carbon bonds. Anabolism, where you build them. You ingest something, you make things, you break it up, you generate energy. That's what you do. Catabolic pathways and anabolic pathways. Well, the CAC is amphibolic. Let's go back a slide. Why? Because it can make energy, it can break carbon bonds, depending on what the demand is. But there's a cost to it, isn't it? If I'm not breaking carbon bonds, I've got not much ATP, not as much energy. If I'm making things like that, I'm not as much energetic. But if I've got lots of those products, I can make lots of energy. What else is there? This overall net reaction. Circle acetyl, 2 carbons. Forget the CoA, that's just the carrier yet again. Some NAD+, 3 of them, some FAD, GDP, and organic phosphate water. What did you make? 2 CO2s. Some of them you're going to use elsewhere, some you're going to exhale. Some NADH, FADH2, GTP in this case, a couple of protons and a CoA. You've recycled that as well. You need to know that for the exam, the overall equation for the citric acid cycle. But for 1 acetyl-CoA, if it was 1 glucose molecule, what would I write next to that acetyl-CoA? 2 times. Boy, that was tough, wasn't it? Do you want to have a quick break now? Catch your breath? First day back after a long week off doing nothing except lying on the beach or skiing down some mountains. We'll start back at 2:45. I know you will have holidays doing nothing. There's nothing to do in the semester break, is there? Get some fresh air, it's a warm day, you'll go to sleep. Because we're about to go into this stuff. Okay. I see Superwoman's back. Now it's time to start. Is that okay?
That's right.
That's right. Alrighty. What have we done? We've ingested things, we've turned them into things, we've taken to the mitochondria, we've broken things up, generated CO2. What are we doing now? We're trying to use those electrons and protons you've generated, because have you made any ATP yet? Not really. Not really. Not enough to keep you alive. And you know you're going to have to transfer these electrons to the ETC, electron transport chain. Now, in the exam, I mentioned there are 60 multiple choice, 4 short answer questions. Short answer, you can use diagrams, schematics, but you must write something. You must convince me, because I'm marking the short answer questions, all of them, that you know something about integrating this information. And at the end of semester's review, I'll tell you the topics you should prepare for, how to get organised. And those that had me in other units know that's kind of important, isn't it? Take note. Okay, let's have a look at the animation for the mitochondria, the ETC set. Here's our convolution. So we're looking at the intermembrane to see what's going on. Still celebrating, obviously. You love it. You totally love it. Let's get back to our mitochondria, shall we? Because he's about to have lots of cake, and what is he going to turn that cake into? Mitochondria or fat? Let's go to our inner membrane. You can see it here. Convoluted, what's in that inner membrane that's rather pinkish? You're going to write down ETC. What have you just done? Well, through the citric acid cycle, you've generated electrons and protons. Let's have a look at this ETC set. Now, before I go on, there are 3 major proteins in there. There's one missing in this animation, complex 2. I've already warned you, it's already embedded in the membrane. I'll come back to that shortly. Here's complex 1. That thing there, that Q, I want you to note is Q, the carrier, not Q, the bond, give me some fancy toys to shoot somebody or whatever. It's the electron carrier. I want you to note immediately, before we get to too much detail, Q is stuck in the membrane. It doesn't go anywhere. It transfers electrons in the membrane. It's not cytochrome C. What have you got here? This complex, complex 1, has got electrons and protons coming at it as NADH. Let's go through the steps slowly. What do I do with those electrons and protons? Please note the sequence. That's kind of critical when we discuss this. There's my electrons and proton carrier. What did I do? I transferred my electrons into this protein. Now, this protein has lots of iron-sulfur centres. It's like the electrical wiring in this room, not copper, but iron. Iron allows electrons to bounce through this protein. As it does, it kind of charges it up. Once it's charged up, it transfers the electrons, there's the blue arrows again, to Q. If I transfer my electrons to Q, it's now got 2 negative charges. What do I need to do? Balance the charge, like all chemistry. I'm going to put some protons on Q, so to speak. What have I done with this complex 1? It's still fired up. Too much raspberry cordial. What am I going to do? Move protons through it. Pump them out. There they go, 4 of them. And I'll come back to this in the still version later on. What does Q do? Well, Q's a carrier in the membrane. It's going to transfer those electrons across to complex 3. Remember, we've skipped complex 2. If it transfers its electrons to complex 3, that's going to zoom through more iron-sulfur centres, so it's like an electrical conduit of iron, as in Fe. What does Q do? Well, now it's reduced or removed its negative charge. It donates the protons to the intermembrane space. Let's watch that happen. Now I'm going to fire through this complex. It's now fired up. What's it going to do? Pump protons through? No. It's going to grab the electrons and transfer them cytochrome. So you go back to that Harvard animation. What happened? You saw a little molecule sit on somebody and light up. That light up is electrons. Cytochrome now is charged. It's in the intermembrane space. It's going to move to somebody else. Who does it move to? Complex 4. But before it does, what have you done to that protein complex? Fired it up to pump more protons out, 4 of them. What does cytochrome do? Finds a mate. That's complex 4. This complex is a bit different. It's going to accept those electrons. Let's watch it do that. A couple of them. It's going to fire up that complex. Do you recall what I'd said complex 4 uses? If I remove complex 4 from your cells, oxygen. It's going to use oxygen and make water. You're a desalination plant, turning oxygen to water, but first you have to charge it up. But you'll notice there it says half an oxygen molecule. That's 1 O. 2 protons, 1 O makes H2O. You breathe in O2, don't you? So you're going to make a couple of waters from 1 O2 molecule. There goes my water. Now this thing's charged as well. How many protons does it pump out? Piddly 2. Why do you need to breathe so much oxygen? Because it's that much more that you need. There's our complex. What's missing is complex 2. That's stuck in the membrane. Who did I say that was? That last enzyme, one of the last ones in the citric acid cycle. What have you done now? You've generated this proton motive force. Because all of these protons on the outer surface of the inner membrane, and because of all the negative charges of amino acids, other proteins, blah, blah, in the matrix space, these characters are desperate to come back in to balance the charge. Can't do it. The only place is through ATP synthase, or I want you to note UCP1. All right, let's get back to the details here. Animation's nice and simple, isn't it? This is just a coloured inversion. What's this complex here? There it is, complex 1. This character stuck in the membrane. What does it do? Grabs electrons and protons from NADH, which you got from the citric acid cycle. It bounces through here. Please note again, the blue arrow through all of this is the movement of electrons. The red arrow is the movement of protons. You've got a balanced charge, but your aim is to pump protons out. This thing is a monster. You see all these shaded pinks? It's a 42 different polypeptide chain protein. It's a huge molecule. It's the first part of the ETC. Remember I said they had iron-sulfur centres? It has 6 of them. Basically, electron jumps from one iron-sulfur to another iron-sulfur to another iron-sulfur. As soon as it gets into the membrane, it transfers the electrons to Q. There's Q. On Q, Q grabs a couple of electrons to balance charge. It makes a couple of protons come in as well. They're stuck in the membrane for a short period. It goes around, and I want you to notice that dashed line there, here from Q, is not going out of the membrane. It's stuck in the hydrophobic domain. You'll see why, because Q is a hydrophobic molecule in a moment. There it is going around complex 2. It can then transfer the electrons and protons to complex 3. What happens there? A couple of protons out, a couple more out, total of 4. Where do the electrons go? To our acceptor. That's what it's called. Cytochrome C is an acceptor of electrons. That thing bounces in the intermembrane space, a bit like a ping pong ball or Australian rules. Just make sure Richmond don't win the grand final. Sure GWS does. Bounces the electrons into complex 4. What do you do there? Grab the oxygen you're breathing in, you're inhaling, make water, pump a couple more protons out. This complex 2 is succinate. We're going to see that in a little while, that dehydrogenase I asked you to circle. Part of the citric acid cycle, taking in electrons through Q, transferring them yet again. Now, in the past, we would go into the details of all of these molecules. We're not anymore. Chews up too much time, and it doesn't help you any more than what this particular schematic does. I'll tell you now, there is an exam question talking about the ETC, the short answer question. You need to elaborate on it. What does it do? For every pair of electrons passed through the ETC, 10 protons are transferred to that inner membrane space. The overall equation you need to know, NADH plus 11 protons, which is N. What do you think N might stand for? Not nucleus, not neutron. The matrix side. See there, the N side, the matrix side of your mitochondria. Where are you passing your protons through? The P side. So that's our equation. NAD plus is what we generate, 10 protons on that side of the membrane and a water molecule. Have you generated any ATP yet? Bloody hell, it's a long path, isn't it? Take glucose and fat all the way through this to generate the ATP to keep you alive. This is essentially a proton pump. Are we done with this one? Are we comfortable with it? Is everyone clear on it? I said there's an exam question, a short answer one. How does it work? How many ET sets are there in your heart muscle versus your normal striated muscle? Would that be different to a smooth muscle, do you think? Where's your smooth muscle normally? Muscle cells. Sorry? Whereabouts? Of what? Of your blood vessel and your gut. Rhythmic contraction. It's not the same as striated muscle. Can I move to the next one? Okay. Who is this guy? The inner mitochondrial membrane is impervious to NADH. I've said it's very difficult to get anything through it. You need proton transporters or different transporters of types. This thing is called the glycerol 3-phosphate shuttle. How does that come into play? Well, what was glycerol doing for you? Do I need to show you again? Those three knuckles are glycerol, aren't they? If I charge it up, I could make a fat molecule, couldn't I? Triacylglycerol. If I cleave all those fats off, what have I got? A glycerol. I can charge it up. Could a cell use that for energy as well? Well, here it is. Our glycolysis through this pathway, there's my glycerol 3-phosphate. What do? The charged phosphate 3-carbon molecule through this thing, a glycerol 3-phosphate dehydrogenase. It's stuck on that membrane. I want you to jot down it's an integral membrane protein. It's stuck on this membrane surface. It's on the outer surface. The glycerol phosphate doesn't need to go into the mitochondria. What's it doing here? Transferring electrons via FADH to Q. What does Q do? Hand them off to complex 3. So that fat, you can cleave off the carbons to make acetyl-CoA. You can also take those 3 carbons to make more energy as well, not just make glucose. That's why this is here. This is essentially a one-way shuttle. Once you cleave that, that's it. It's all over. Electrons and protons are going into that pathway. That's done. Hence why I've dashed that red line around that FAD process here. Doesn't generate much energy, so a glycerol is pretty weak in terms of energy outcomes compared to fats and glucose. But it's a way to use that glycerol moiety to generate energy. Where does it occur? Well, skeletal muscle, I want you to write down when the demand's really, really high. Why? Because that thing is in a fat cell next to you. We're going to see that in the next few weeks. You're going to send out each one of those fats, then you send out glycerol. Some of it goes back to the liver, but sometimes the muscle can grab it. But then you're starting to get low on blood sugar. What happens to your brain? Ketone bodies and this stuff. I can use some glycerol in my neuronal cells as well. Can I move to the next one? Comfortable with this one? All right. I mentioned complex 2 was part of the ETC set, but it wasn't an animation. Well, here it is now. This thing, succinate dehydrogenase, right underneath it, part of the CAC. Let's go back and check where it is. It's not there, it's not there. Here it is here. It's in the second half of the cycle. What are you generating from this? FADH2. Just right next to that, go back to that slide, integral membrane protein, part of ETC set, complex 2. sorry, this one here, succinate dehydrogenase, integral membrane protein, complex 2, also known as, aka complex 2. Can I move back from this slide now? Yep, okay. Where are we? Now this one I'm showing a bit more detail. I mentioned those iron sulfur centres. This one's kind of useful because you can see in here this motion through these iron sulfur centres. You notice the blue arrow, remember that's the movement of electrons. So it jumps from iron sulfur to iron sulfur to iron sulfur, there it is, moves off to Q. Q is term ubiquitinone. We're going to see that in the next slide or two. Hydrophobic molecule stuck in the membrane. It's an acceptor of electrons and protons and a donator of electrons and protons within the membrane. This thing, you can see all those coloured components there, has multiple subunits and prosthetic arms as well and legs. It has a heme function like we saw at the beginning of the semester. It has 5 prosthetic groups, 2 different subtypes, 4 different protein subunits. But it's one big enzyme complex. It does 2 things, as I have down here in italics. It converts the succinate to fumarate, circle the citric acid cycle to keep that going. The other thing it does is it captures and donates the electrons to the transport chain, the ETC. Final exam's looking a bit scary now, isn't it? Oh, really? Okay. I remember that. Yes, you're right. But if you look at those guiding questions, are they important? For someone who did another unit with me or still run those kind of things, I would strongly recommend you look at those. And particularly read the pages in Leninger. It will help you digest it. We had a question during the break. You know, that mid-semester exam was a bit challenging, wasn't it? Good, because as I said, it's a formative assessment. It's there to challenge you. You're not going to get the same kind of questions. Indeed, you won't get any will assay questions in the final exam. No calculations in the final exam. Oh, there's a sigh of relief, aren't you? Doesn't mean I'm going to mark it any easier. Are we done with this one? Yes? Oh, yeah? Now, before I move to the next slide, I just want you to keep in mind the electrons wind the protein up. It's like raspberry cordial. Then the protons pump through. So I'm going to be checking on that in the exam. Now, the protons don't pump through because of the electrons. In that sense, the protein's wound up because of electrons, which is important in a moment because too many electrons mean reactive oxygen species, means damaged DNA, damaged mitochondria, tumours. Dead, dying cells, mutations. You've got to manage these electrons. Can I move to the next one? Yes? Okay. Here's Q, ubiquitinone. There he is there, a nice simple ring structure. What does it accept? Electrons and protons. You'll notice the schematic showing it within the lipids of this membrane. Little balls are the polar groups. There's the long chain fats zooming through the membrane. Stuck in the membrane, not getting out. It's not cytochrome C. I see that confused in the final exam. Cytochrome C is a cytochrome. It has a colour. That's why your mitochondria are dark, particularly. It's bouncing electrons out in the intermembrane space. You can see everybody with these blue arrows transferring their electrons to Q to try and contain them in the membrane. What's Q going to donate his electrons to, or her electrons to, or its electrons to? Which complex? Complex 1, 2, 3, or 4? 1, 2, 3, or 4? 3. Remember, 3 then donates into cytochrome, then goes to complex 4. I'll be checking in the exam. This benzoquinone, obviously hydrophobic, moves pretty freely through all this stuff. It's like that little fish in Nemo, that's the one. Through those stinging coral thingies, whatever. Anemones, thank you. Or enemies, one of the two. Very freely with its electrons. Does it have protons when it has electrons? If Q has electrons, does it have protons? Yes, you have to balance the charge, remember. Okay, can I move to the next one? Oh boy. Now, when you get to the exam, think of James Bond, because then you'll think of Q. What did Q do for James Bond in most of the movies? Gave him something, didn't he? All those toys to do something with. Don't forget Q. Many people do in the final exam. And if you write down James Bond, you'll get a LOL, won't you? Can I move to the next one? All right, I tried to scare you with reactive oxygen species. It's a real phenomenon. It's not something to be taken lightly. Now, this happens particularly, I want you to take note, happens particularly when you're on a high carbohydrate diet and you're not exercising, repeat, not exercising sufficiently, and you're still breathing a lot. What are you doing? Lots of carbons, lots of cleavage, lots of fat deposition, lots of electrons running around, lots of reaction oxygen species, but no utilisation. So we see more ROS species in more sedentary individuals that are on a high carbohydrate diet. What does that induce? Diseases like obesity, diabetes, cardiac issues. It goes on and on. I need to scare you because, as I said to your colleagues at Hawkesbury, and also again next few weeks, it's now getting down to 1 in 3 obese in this country. Children become more obese. It's long-term defects because this damages mitochondrial DNA, damages the mitochondria, means your lifespan's reduced as well. So exercise needs to be balanced with diet all the time, not just when you feel like it. Even when you're a student, you should exercise. I know it's difficult. Let's look and see how this works. Well, here is your ETC set. There's complex 1, complex 2 is missing, complex 3, complex 4. What are they doing? They're generating Q, a highly charged molecule. Lots of electrons and protons. It's a phenolic group in this case. What are you doing with Q? Well, any oxygen running around with it, what are you doing? Inhaling oxygen. If you're not using that oxygen to generate ATP ultimately, it's going to be going to use to make this stuff free radicals. Circle that -2 as a free radical. That thing, through the dismutase, the superoxide in your cells, makes peroxide. That's damaging too. These will cause damage to your mitochondrial DNA and the function of these proteins. What does your mitochondria try to do? Well, we've seen this character before, haven't we? Glutathione. Remember we said it was stuck together? What do? Break the disulfide bond and you generate this reaction. We saw that earlier in the semester. That process allows you to convert hydrogen peroxide, which is a bit nasty, back to water. But you need the glutathione. How are you doing that? Well, you're doing that with some of these reactions with NADPH. This overall reaction allows you to generate the glutathione, this particular character here, running through this active and inactive enzyme, which is termed oxidative stress. Now, you've all heard of ROS species, haven't you? Yes, some have, some haven't. These reactive oxygen species, which I've highlighted down in italics for the rate of electron entry into this respiratory chain. That's the ETC. And their transfer of electrons to the ET, if they're mismatched. Too much oxygen, not enough workload, you generate these free radicals. Here it is, this superoxide free radical. The production increases at complex 1 and 3 particularly. Too much of that, you need the dismutase to try and reduce its free radical nature. Becomes an issue for you. You might need some sulfur functions then, that glutathione, to manage them, which we're going to see in the next couple of slides. This is an age-dependent effect. This generates more mutations in D and A mitochondria the older you get, particularly if you're more sedentary. Feeling good about being young? Good. Won't last long. You'll be old soon enough, just like me. Can I move to the next one? No? Have I scared you enough yet to exercise and watch your diet? Not yet? There's still time. You wait till we talk about fat deposition of foaming macrophages. Can we move on now? Everybody else right? Yes? Oh, boy. Complex 4. A little bit big, 200,000 odd molecular weight. What do we say was unusual about this one? It's got copper in it. Copper. Some of you have had Chris Jones, maybe heard about him. He works on copper in cells. Also works on things like amyloid plaques. Copper's involved in those. You heard about amyloid plaques? You know where they occur? As you age, they tend to accumulate in your brain. You get brain dysfunction. As you age, your brain shrinks. As you age, you start getting things like dementia and so on. Unless I've tested you in metabolism and you're very smart so that you can last longer with a big brain. Think of it that way. What does this stuff do? Well, here's the copper centres. It's assisting with our oxygen to generate what? Water. When you charge up this molecule, here comes the charge from cytochrome C, transferred to copper, charges up the molecule through those blue arrows. In comes the oxygen you've inhaled via your red blood cells. Out goes water. You then use some of the protons. Now that's the key thing with this particular slide. Not all the protons you generate are pumped out. Two of them are going to use to make a water molecule. Yeah? Make sure you write that down, because in that short answer question, that's going to be clearly important, isn't it? Not every proton is sent out into the matrix space. It's here to make water. What are you doing? Taking oxygen, turning it into water. What are you also really doing? Well, you're getting rid of those free radicals by turning the oxygen into water instead of a reactive oxygen species. And please note on your slides or on your paper, on your computer, we're not the only ones with Ross species issues. Every species that has a mitochondria or a chloroplast has the same issue. Plants do, microbes do. Trying to manage them. Which microbes? Yeast particularly. You'll notice also it has some heme functions in there as well, so it's a bit of a copper iron centre. Does that mean you're going to go around and lick all the copper wire in your house somewhere? No. But it means copper's important to keep the ETC set working correctly. Copper's part of your diet. Just in case of interest, we now have a copper fluorophore. We can actually see how much copper you have in these cells, in a live cell. Can I move to the next one? Is that a yes or not sure? Just a second. Sorry? Yeah, every species that has mitochondria has an issue with reactive oxygen species, so including yeast, because they have mitochondria. Okay? And they're a eukaryote, hopefully you recall that. Okay, time to move on. Yes? I I'm going to make an executive decision and move on. I want to get to this guy. Now, this is ATP synthase. Now, unfortunately for you, the schematic is upside down. So is the animation. Before I take you through the animation, down here, this is the intermembrane space. Up here is the mitochondrial matrix. Please note, down here is the intermembrane space. What are you doing? You have lots of protons running around here trying to get back in. They can't. So right along here, H+, 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 lots of them. You've just generated them. What do you have on this membrane? Lots of net negative charge. Right down, right, minus, minus, minus. What's happening? This guy has to come back in. He can't get through the membrane. He's got to come back in through this water wheel. That was that beautiful Harvard animation, that rotating protein. And I don't just say that because it's beautiful animation. We can see that. We can, molecular-wise, put a fluorescent tag on this thing and watch it rotate in cells, moving around like the animation's showing you. This thing is called ATP synthase. It has an FO domain. Think of that as the water wheel. Protons come through, it spins. As it spins, it rotates this shaft just like you have in your engine of your car. You know the main... No, you don't. I didn't think so. That shaft running through your engine is offset. This shaft running through here, this gamma domain here, is slightly offset. As it rotates, it squeezes and contracts this domain called the F1 domain. I want you to note this is where you generate, I'll get this thing out of the way. This is where you generate ATP. Now this ATP synthase is an enzyme, but it's an unusual enzyme. It's not an enzyme where it has a catalytic site and does a particular reaction. I mentioned a few times it's an enzyme like a vice. It grabs ADP, inorganic phosphate, squeezes and squeezes until pop, it's made ATP. The squeezing is this shaft rotating. These B delta domains are simply holding it together like your motor's holding that shaft in the middle together. Please note this area here is in the matrix. You're generating ATP in the mitochondrial matrix as protons come through, which is what this animation is you're seeing here. Excuse me. Let's watch it. There's all those protons. Intermembrane space, what are they coming through? The water wheel. Every time you turn, you make an ATP. But please note, there are 3 domains, the alpha and beta domains. Each one of those domains is going to generate ATP. So there's 3 ATP sites. Let's have a look at them. There's your water wheel for protons. Now this animation is not quite as consistent as the schematic. Let's wait for our protons to start moving. They better move soon or we're dead meat. Now this animation is showing, oh, they're just spinning through here. No, they're not. They're actually winding around and spinning the wheel. Keep that in mind. This is this schematic showing. The protons literally spin the wheel. They spin the axis. In they go to the matrix, they've just neutralised the charge now. What happens to your... Just stop that. This is the side view. Okay. Looking at the side, there's the head domain, the alpha-beta domain. I'm just going to turn it around now in the animation. You're looking down on from inside the mitochondria to that head domain. Let's get there. Where's my damn mouse? We can see the gamma shaft in the middle. You can see that it's offset. You'll notice the beta, which I said here, there's 3 sets of them. The alphas keep them together. Each one of these sites is a site to grab ATP and organic phosphate and make ATP. Watch the rotation. A full rotation does what? One side's open, one side's clamped, one side's ejecting. We do a rotation. What do? Change the other two at the same time. Squeeze up one, release one, clamp one. Continue the rotation. What are you about to make? ATP. What did you do at the other two sides? One's being released, one's being grabbed. So a full 360 rotation takes in one, clamps one, and releases one. Make sense? Keep doing it. That's all this thing does, which is what the steel version is you see on the top, the rotating shaft in the middle. Now I'm showing you a bit of enzyme kinetics. Remember that stuff in FP&G where you had activation energy and all that stuff you want to forget? Well, you're probably used to seeing something like this, change in free energy, kilojoules per mole, or maybe you're trying to forget it. What we have down the bottom here is time. What does E stand for? Enzyme. What does S stand for? Substrate. A normal reaction, now there's variants of them, you often have to get over the activation energy to generate an enzyme substrate complex and then make the product. That's what you have here. When you're finished writing, I need you to look at this panel here and note the difference for ATP synthase. Most reactions, not all, I grant you, require activation energy to generate an enzyme substrate complex where the two come together. You get over that hump, you make a product. Get up to it, force it together, make a product. Are we ready to look at the second panel now? Yes or no? This is ATP synthase. You've just seen the reaction. Well, let's just focus on one of these sites. What do? Incoming ADP inorganic phosphate. A little bit of energy, not much. I squeeze them up. There I have. I've just generated ATP. That didn't take much work. The hardest part is kicking the little bugger out. Out you go, off you go. That's the biggest activation energy required to spin that shaft, to get rid of it, which is why it's a bit unusual in that sense, not like most of the enzymes you've seen before. Is that part of the exam question? I think I'd talk about it if I was you, if you want the marks. So the major energy barrier is not generating the ATP, it's getting the ATP out of this ATP synthase. Which complex component of it, the FO or the F1 domain? What's the FO? Oh, I'm circling around. F1, I've just made something. And I need one full rotation to make an ATP. Whereabouts is this? In the matrix. So I've got mitochondria generating ATP in the matrix. I have to get that out. That's going to be important in the next few weeks. Are we comfortable with this concept? Okay, it's an integral membrane protein. It literally rotates. What I find beautiful is the design. Now that's nature designing a protein that spins like an electric motor. They've already done it before we even got close. There's no graphite, there's no bearings, there's no magnetic pulses. It's just amino acids against amino acids allowing rotation. How many of these do you have? Well, I want you to jot down one of these is hooked up to every ETC set. So every ETC set has an ATP synthase associated with it. If you have 10,000 ETC sets, you've got 10,000 ATP synthase minimum. Yes, mate?
Is one ETC set in line next to each other, or can there be other complexes in between it?
Good question. Is one ET set's kind of in line? Essentially, yes. They're kind of not so much in a line like the schematics, they're kind of grouped together. It's a little bit like enzyme channeling. It's not like I hand it off here, it takes a while to run over there to give it to complex 2, 3 or 4. They're actually associated. So when you look inside the mitochondria, you see them aggregated together with an ATP synthase. It's like a close-knit family. Does that answer your question? Okay. We comfortable with this one? Yes, mate? What do you think? If you look at the delta and the alpha, and even the epsilon down here, what do you think they might be doing? If you had an electric motor just with the shaft and the cable, and no housing, what would happen? Fall apart. So there's structural proteins keeping things together. Okay? Stabilising it. Particularly this bit that hangs out to the side, it's like that housing for an electric motor, which I know most of you haven't looked at, but that's okay. Can I move to the next one? Alrighty, bless you. We're almost done. Now what's coming in and going out? What did you start with? ADP and inorganic phosphate. How does it get into the matrix? There's your substrates. What's your product? ATP. You've got to get it out of the mitochondria. Here is our mitochondrial membrane, the inner membrane. This thing here, what does that look like? ATP synthase. I want you to jot down FO, F1. Which one's which? Let's look back here. F1 is the ATP maker. FO is the water wheel stuck in the membrane. What have I got to do? Well, first protons come through, but I've got to have some inorganic phosphate get in there. What do I need? This thing, a phosphate translocase. It's an integral membrane protein. It's called a symporter. Why? Because when a phosphate comes in, so does a proton. Uh-oh. Now we have a problem, don't we? We pumped all these protons out, now someone's coming back in just to get phosphates in. So all those we've pumped out, not everyone's going to be used to make ATP, are they? Through ATP synthase. Please note that. Not every proton goes back in through ATP synthase. Some comes in via this thing, this symporter. What do you do with this character? Use it to make ATP. What else do you need? ADP. This thing is called an antiporter. This integral membrane protein, the adenine nucleotide translocase, takes that ADP, transfers it into the mitochondrial matrix through the inner membrane. But if I've got one in, because it's an antiporter, one's got to come out. Who comes out? ATP. Does that make sense? ADP in, ATP out. ADP is your substrate, ATP is your product. Where does it go to? Inner membrane space, cells use it for all sorts of things. Charge up molecules, get them busy doing their work. Now this process is basically fluxing across the membrane. There's one we're missing though, isn't there? I told you already brown fat generates heat energy. We're ready to move on to that? Is that a yes or a no? Okay. What have you just made? It's an energy carrier, isn't it? Not the only one. We're going to see creatine and creatinine later on. They're just kind of buffers for phosphate, particularly in muscle. What do you see here? A base, a sugar, and I think 1, 2, 3 phosphates. What do you do when you're activating somebody? Clip a phosphate up. If you want to activate somebody else, clip another phosphate. You get down to the monophosphate, what do? Go back to work. This is your energy currency. Now, generally, you've got other characters as well involved, things like magnesium and manganese. How many times have I mentioned those characters? For work, they are important cofactors. What does ATP do? Well, it contains the energy in these phosphate bonds until somebody uses them. Who uses the phosphate bonds? What kind of enzyme? Kinase. Please write it down. There's all my ATP. What do I need? Phosphorylate somebody. What do I do it with? A kinase. Now, I want you to just cast your minds back to glycolysis. Remember, there's a couple of kinases there, but did they make phosphorylated proteins? Bottom part of glycolysis, what do they make? ATP. ATP, remember that, it's important to contrast. My favourite question, compare and contrast. What's the similarities, what's the differences? We done with this one? So yes or no? Yes? Cool. Made a decision for you. All right, now we've got your fat. your brown fat particularly. Here is a white fat cell schematic. That big yellow thing is lots of, write down triacylglycerol. We saw molecules activate that. What did they do? Things like perilipin, activating enzymes to cleave off individual fatty acids, remove the glycerol. Fatty acids disappear into the blood carried by somebody. This thing doesn't have many mitochondria, that's why they're called white fat. Nucleus is squeezed off to the side. You fill up that fat droplet, too much, tells the cell to divide. You have more fat cells. I said before, you're born with a number, you're generally consistent with that until you tell them to divide. Once they divide, you have that number of fat cells. They increase in size as well. But your brown fat's different. I mentioned before with our birthday boy, it's down your back, around your kidneys, keeping your kidneys warm, your liver warm, especially when you're born, and now, coming out of winter, keeping you warm as well. You'll often hear people, at least the old people like me, say, oh, you've got lots of brown fat, you know, you're really busy, really active. During winter, you know, you don't put many clothes on. Well, as many thick clothes, put it that way. Why? Because you're generating lots of heat energy. Now, be careful with that, because after we've already done semester break. In a few weeks, we're going to talk about things that change your metabolic rate, like thyroxine, T3 and T4, coming from your thyroid, which increases metabolic rate, generates more heat energy. These characters have a lot of mitochondria. That's those little squiggly bits in there. They don't have one big fat droplet. They have lots of them. Why is that? Because they're going to break up little bits and generate lots of nice, warm energy for you to keep you warm during the colder periods. How do they do that? Well, here is our membrane from our mitochondria. In here, is this the inside? Yes, it is. That's the matrix. So we've gone from one side, other side, now we're vertical. So right down along here, you're going to write lots of H pluses. Along here, you're going to write lots of negatives. What have you done? Well, there's complex 1, 2, 3 and 4, busy pumping protons out, doing all their work, making ATP. But in your brown fat, when you need it, some of these protons are not coming back in through ATP synthase. Look at the board. Here's ATP synthase coming back in. Some of them came back in through this character. Oops, where is he? Through here to help make our substrate. What have you got here? This thing under thermogenin. You need to know that name. Underneath you're going to write UCP1. Why is it called UCP1? Not the stuff you used to hear when you were a kid, ICUP, or eat all the peas on your plate. That'll help you remember. Uncoupling protein 1. It's uncoupling the protons from ATP synthase. This is a little bar magnet. As the protons come through thermogenin, hence the name, also known as UCP1, it generates heat energy. It keeps you warm. This is a protein made how I showed you earlier, stuck in the membrane, starts in the rough ER, that membrane is moved to the mitochondria, that membrane now can be used to generate this energy process. That comes from a gene. Now, that particular gene is often related to maternal genes. Most of your mitochondria are maternal genes. We'll talk about in a minute. Where is that mostly in? Brown fat. You will generate more gene activation, that is RNA for it, messenger RNA. You'll turn on it when? When we came into winter. Cold months, or it's really cold for a start? No, it's not. You're just not readily adapted. If I take you down to Antarctica, what's going to happen to you right now? You will, you'll rug up a lot, but after a while you'll start to adapt, won't you? A couple of days, a couple of weeks, you'll eat a lot. And do those people working in those environments generally gain weight? A little bit. Fat insulation, that's normal. One of my technicians from Syro went down there for a while to take some seal blood. But then you adapt by generating more UCP1 protein in your brown fat. What's that doing? Keeping you warm. You don't have to rug up as much anymore. You've adapted. But if I bring you back here to a hot summer in Sydney, you'll feel it the first few days, won't you? A few days later, maybe a week or two, well, it's not so bad now. It's not just whether you peel off, it's you've got less of this protein in that cell membrane. mitochondrial membrane. Does that make sense? You induce gene expression and you reduce it depending on your environment. Can I move to the next one? Okay. I have to say this is a pretty critical slide. There's a few key characters on here you need to know, but this slide summarises things. Remember that ugly grey slide I started you with? You need to fill in the gaps, put in the bits and pieces. This is another one of those slides. If you know this slide, you know the grunt biochemistry of metabolism. Whereabouts? Well, this is the mitochondria. You see the outer membrane. This is our inner membrane. I'm going to let you write and I'm going to talk about it. But please note the bits on the slide I've given you on views and the bits you have to fill in. That's really important. Look carefully, keep looking carefully. I need you to. Now, if you get to the final exam and you think you could schematise this thing in the pages that I give you, I'm giving you one line page and one blank page in the exam booklet. Not an exam booklet you know of without the questions, the exam I've set up. If you want to draw this thing, that might help you a lot. Do you need any more hints? We ready to talk about this one? Is that a yes or a no? Chris isn't ready yet. It's like one of those dogs in the back of the car, you know? Are we set now, mate? Okay. You guys right? Why is this slide so important? Because it summarises all of the key pathways in the mitochondria. We've built up to this. This is the crescendo of the grunt biochemistry. What have we got to do? Well, we generated pyruvate from what? Glycolysis. I said we had to get it into the mitochondrial matrix. What have you got to use? In that mitochondrial matrix, you've got to use PDH. Write it down. It's missing, isn't it? PDH to generate what? Acetyl-CoA. There it is there. But to get pyruvate in, I need this protein. That's only just been found recently. That yellow thing is mitochondrial pyruvate carrier, MPC, not UPC. We knew it was getting in, we just didn't know who did it. Finally found. There's my pyruvate. What do? I activate it with CoA. There it is now with PDH. Write that down there. There's my acetyl-CoA. I can enter the citric acid cycle. That's all it is, just a linear form. What do I produce? CO2, lots of electrons and protons. The ultimate outcome of that is OAA. Now down here you see fats. Remember we needed a shuttle, didn't we? Activate them, put them on carnitine, transfer them through, then put them on another acetyl-CoA. That's what you're seeing here. Activate them through that process, new CoA, now we've got this CoA molecule. I break up my fat. What's that process called? Long chain fat, write it down, beta oxidation. Every time I cleave a carbon bond, more electrons and protons. But what do I produce? Acetyl-CoA. Can that enter the citric acid cycle? Yes, it does. Could I drag it back out through citrate to make more fat? Who would do that? Muscle cells make fat? No, just the liver. There we are, running through the citric acid cycle again. Keep in mind there's all these protons building up on the outside of our mitochondrial matrix. Down here you see this box, box number 2. What would you call that? ETC. What was missing on this box is what I've drawn in, cytochrome C. What does that bounce from? Complex 3 to complex 4 with electrons. What are you doing here? You're taking in oxygen generating water, but you're moving those blue arrows right down that blue arrow's electrons. Red arrows, the protons. I've done all that. Here's all my protons out here. What happens? They come back in through ATP synthase. Which is FO and which is F1? Indeed. What do I need to get in there? I need substrates. I need inorganic phosphate, ADP. There's my transporters. That would be most cells, wouldn't it? Yes. What about this character, UCP? Which cells would that be? Circle it and write brown fat. Nobody else is doing that. Not the liver, not white cells, not muscle cells. Brown fat, UCP1. This is an encompassing schematic. I've told you there's a short answer question on the ETC. If you can draw this schematic or get close to it, you're pretty close to getting that exam question sorted, aren't you? You finished with this one? Yes? Okay. Let's summarise that. I don't mind if you use all the acronyms we've used all semester, as long as they're the right ones. The electron transport chain, that'll save you writing out that word every time, okay? And you don't have to say electron transport chain bracket ETC. I know what you mean in an exam. Save time. Reduce cofactors, pass these electrons into the electron transport chain in the mitochondria. Which membrane? The outer or the inner membrane? It's a stepwise transfer of electrons accompanied by directional transport of protons. What happens first? Electrons get busy, charge up the protein, then the protons get pumped out. That's pumping out against the gradient. You're moving a positive charge, even though it wants to come back in. That's why the electrons are required, to pump the protons out. That generates your electrochemical gradients, the proton motive force, which is going to drive ATP synthase. It's going to be helpful for UCP1. What else is it helpful for? Bringing in inorganic phosphate. Remember, an inorganic phosphate, to make ATP, has to come in with a proton. Our muscle and fat cells, well, they're using UCP1. Now, muscle doesn't do it so much, I'm afraid. It's a bit of talk in the literature. Depends on the species, I'm afraid. Here you're seeing a nice little cell micrograph showing you this brown region. What does the brown region mean? That's a stain for this case, mutated cytochromes. As you age, your mitochondria experience more and more reactive oxygen species and start mutating the mitochondrial behaviour. They become less active. Here is a bunch of tissue. This tissue might mean not every cell has the damage. Not every cell is doing the workload at that level. But as you age, you see more and more damage to those cells. Does that kind of make sense? Think about old people, older than me. What happens? They lose protein mass, don't they? It's much harder for them to work. Brain function declines. All of those things that age does to you. Now, if you have a lot of carbohydrate diet, not much exercise, that's going to increase the rate of aging because of the damage to the mitochondria. I mentioned before, this is maternally inherited, so your parents, your mother particularly, is donating the mitochondrial genes to make those mitochondria. If you do advanced cell with me, as a couple of you have, you know that we can do mutations and we can actually transfer mitochondria from a donor with healthy mitochondria into a recipient. There are some individuals in this world who have somebody else's mitochondria, but their parents' DNA. Because lots of DNA damage occurs in mitochondria. Because remember, they have their own DNA, don't they? Are we done with this one? Are we done for the day? Almost. Before I let you go into it, let me remind you, next week, I will be giving you some feedback on the mid-semester exam. Next week, I'll be giving you some feedback on your report, Part A. I'll call it interim feedback while the markers are still going. Thursday this week at 1 o'clock, you will see the list of topic Part B available on Views. You peruse those at your leisure and then you'll select them next week in the lab. Is that clear? Can you also be clear that we're going to mark that very quickly because that's due really late in the semester, isn't it? Yep. Don't expect a lot of feedback from that because we have to mark that quickly and I have to mark the exam. So you need to take note of the feedback I give you next week to incorporate that into Part B. Is that clear? Are we ready to do this thing? ETC is too easy. Sorry? UCP1's too easy. Yeah, too easy too. Now, if you were carrying electrons to complex 3 to complex 4, what acronym might you use? CYT, off you go, all caps.
Capsule?
I'll remind you again while you're doing the quiz, there's another catch-up in week 14. Week 14, where are we? Face-to-face or on Blackboard Collaborate? CYT. All caps.